And the cell will distend or will enlarge. It will swell. The cell will swell to a point where it will finally burst. Now, if you look at the structure of an animal cell is covered using the cell membrane. It does not have a cell wall, as in the case of a plant cell. So because it is only having a cell membrane, which cannot withstand that pressure or that force, the animal cell will eventually burst, and it will release the contents. Now, we also went ahead and looked at what we expect to see when this similar animal cell is placed in a hypotonic solution. Now, a hypotonic solution is a highly concentrated solution. Now, meaning if this animal cell is placed in another solution which is highly concentrated than it. When an animal cell is placed in another solution which is highly concentrated than it, then this is what we expect to see. Now, the animal cell will lose the water. The animal cell will lose the water. Or in other words, water will be drawn out of the animal cell by osmosis. Now, we're talking about the word osmosis here because osmosis refers to the movement of water molecules or solvent molecule from a region of its high concentration to a region of its low concentration across as an impermeable membrane. And that is what we defined osmosis as. So basically, if you're talking about an animal cell that is placed in a hypertonic solution, it means that there'll be, we assume that there'll be a high concentration of water molecules inside the animal cell as compared to that particular hypertonic solution. And for that matter, water will be drawn out of the cell by osmosis. And as you can see, the final appearance of this cell at this point in the final diagram here, the cell shrinks or it wrinkles and it becomes crenated, and that makes me talk of these two important terms that we looked at. One, we looked at what is called hemolysis. And we also looked at another term is called crenation. Now, hemolysis refers to the bursting of an animal cell when it is placed in hypotonic solution. That is hemolysis. Now, crenation is the opposite of hemolysis, and the crenation refers to the shrinking or wrinkling of an animal cell when it is placed in a hypotonic solution. And we have seen what leads to either hemolysis or crenation. Okay, so if there are questions in the area, or maybe if you haven't understood, if there are questions in that area, or maybe you haven't understood, you please let me know. You can directly contact me because I know a good number of the students have our contacts up to this point. You can directly contact us, ask your various teachers, or you can try out and reach me because I'm always available and I'll be able to assist you. Okay? Thank you. So allow me, without wasting much time, to proceed to our today's area of study, and that is we will be able to look at this point water relations in plant cells. Water relations in plant cells. Now, I believe there is no questions about water relations in animal cells that requires an urgent explanation before we proceed. Now, I remember in our last class, I gave out some work which I expected you to between Monday last week and today. So the reason why I gave out that work was so that you could be able to maybe look at some of the terminologies that you're going to meet today and maybe have some rough idea of what they are, so that in our today's class, they don't become a challenge. But all in all, I know that those students who did the work and others, as usual, will never do the work. So to those who did the work, I think you're going to benefit a lot because you'll find the terms very familiar to you. And for those who never did the work, we will keep on praying for you. So water relations in plant cells. This is where we're starting from today. And I would first want to show you these diagrams, and that is why it is my first slide here. So if you look at these diagrams here, they show what will happen to a plant cell when it is placed in different solutions. And our first solution here, we look at what happens to a normal plant cell when it is placed in a hypotonic solution, or just distilled water. Distilled water or pure water is an example of a hypotonic solution. So we look at what happens when a normal plant cell is placed in distilled water or pure water, or what we call hypotonic solution. So when we know that, first of all, it is important for us to understand the structure of an animal cell, of a plant cell, sorry, the structure of a plant cell. And if you look at the structure of a plant cell, it is a bit different from that of the animal cell. In other words, the plant cell is made up of one, the outer covering, which is called the cell wall. The cell wall. Then just beneath the cell wall, we have got the cell membrane. Talking about the cell wall, it is important for you to understand that the cell wall is permeable. It is permeable. In other words, it allows anything to pass through it. It is fully permeable. While the cell membrane is semi-permeable. In other words, it will allow for selective movement of substances to pass across it. So, the cell wall, as we know, is strengthened by the deposition of cellulose. So we talk about the cellulose cell walls. So the cell walls are made up of cellulose, and therefore they are very, very strong. So if this okay, inside the cell, the plant cell, we have got the cell vacuole. We have got the cell vacuole, which contains what we call a sap, the cell sap. The cell sap is a solution which contains dissolved sugars and salts. And for that matter, there is a high concentration of solutes inside the plant cell at the vacuole where we have the cell sap. I will repeat that point. Inside the plant cell, we have a vacuole. And this vacuole contains the cell sap. The cell sap is made up of solutions of salts and sugars, which therefore makes it to be highly concentrated. Are we together? I believe we are. Great. So this plant cell, when it is placed in a hypotonic solution, look at this diagram here. The next diagram that you're seeing from, I believe you can all see my cursor here. I believe you can all see my cursor here. So if this plant cell is placed in the hypotonic solution, water will be drawn into the cell by osmosis. In other words, water will move from its region of high concentration to its region of low concentration, which is right inside the cell. So we are saying that water is drawn into the cell by osmosis. Great. So once water has been drawn into the cell by osmosis, the cell will swell. In other words, the content of the vacuole will swell, it will enlarge, or we can say it distends. To distend simply means to swell or to enlarge. So the content of the cell will increase, and as it increases, this increase in the content of the cell pushes the cytoplasm outwards, and the cytoplasms finally pushes the cell membrane towards the cell wall. So you can see that enlargement in terms of the size of the cell, and that is what has been shown in my diagram here. So we are saying that the cell swells, increasing contents push against the cell wall, exerting a pressure that we call turgid pressure. Now, I'll be able to show you, or we'll be able to learn more about the turgid pressure just after this. Now, turgid pressure is that pressure that is created when the cytoplasm, or in other words, the content of the cells, push against the cell wall. turgid pressure. Okay, so you can see if you look at them, we have got this point towards the outwards. That shows that there is that force, or there is that pushing of the cell contents are pushing towards the cell membrane and the cell wall there. Now, another pressure, now as the turgid pressure increases, or as the cytoplasm and the vacuole contents they push towards the cell wall. Now the cell wall develops a force which will act on the opposite direction to that of the turgid pressure. Now this force is equal to the turgid pressure, and this force, we call it the wall pressure. Now, just after these diagrams, I'll be able to explain more on the turgid pressure and the wall pressure. So just understand that that force that is exerted by the cytoplasm and the vacuole that pushes towards the cell wall is called the turgid pressure. And the turgid pressure in turn develops another resistant pressure or force which is equal and opposite to that of the turgid pressure. And this is called the wall pressure. Okay? Now, I want you to note that when this happens in a plant cell, the plant cell will swell, and finally, it will become turgid. It will become turgid. Something that is turgid is something that is very strong. Something that is very strong. Now, like I remember there's a day I was having some of my students and they were telling me that, teacher, can you see them flexing? Flex. When you flex, you're trying to show me that you're very strong. Now you're trying to make your muscles very turgid so that you be very strong. Now we are saying that something that is turgid is something that is very strong. Now the cell, the plant cell becomes at that point when the turgid pressure equals the wall pressure. And so it is important for you to note that when this happens in the plant cells do not burst. And the reason why plant cells do not burst is because of the presence of the cell wall, which is very strong, the cellulose cell wall, which is very strong and does not allow plant cells to do what? To burst. Now, this is an exact opposite of what we saw while we were looking at the animal cells. And I just mentioned earlier on that the animal cells, they burst when they're placed in hypotonic solutions. And if you look at the structure of the animal cells, you notice that they do not have the cell wall. They are only made up of the cell membrane. The cell membrane cannot withstand the pressure and therefore it will burst. But for the cell wall, it can withstand that pressure, and that is why it develops the wall pressure, which is equal and acts in the opposite direction. So it becomes turgid, but it does not burst. Now let us look at what happens when a normal plant cell is placed in a hypotonic solution. And that is explained or represented in the set of diagrams that you just see here. I believe you can see where I'm running my cursor. If you can see this point, this is what is explained there. So we are saying we are talking about a plant cell that is placed in a hypotonic solution. Now we'll try and move very fast. If you look at this diagram, the first one is talking about a normal plant cell placed in a hypotonic solution. So a hypotonic solution is a solution that is relatively more concentrated than the than the plant cell. So if you're comparing those two solutions, the solution inside the plant cell, we know it is concentrated, yes? But now if it is placed in a more concentrated solution, now we say that the more concentrated solution is now the hypotonic solution. Because if you're comparing two solutions, the one that is highly concentrated is the one that we call the hypotonic solution. So if a plant cell is placed in a hypotonic solution, the plant cell will lose water, or we say that water is drawn out of the cell by osmosis. And what happens once the water has been drawn out of the cell by osmosis? The cell membrane and the cytoplasm, they pull away from the cell wall. And that is shown using this diagram here. Now, if you see where we have got our cell membrane, the cell membrane is pulling away from the cell wall. The cytoplasm is also pulling away from the cell membrane. Now, it's like some kind of shrinking, okay? So now it's losing the shape. It's losing the shape. Now, if you look at the next diagram here, once water has been drawn out of the cell by osmosis, and once the cell membrane and the cytoplasm have started pulling away from the cell wall, then we say that the cell becomes flaccid, or the cell has been plasmolized. Okay, the cell has been plasmolized. Now, I remember in one of the assignments that I gave in our last class, I told you to go and find out the meaning of the word plasmolysis, the definition of the word plasmolysis. Now, plasmolysis is the process by which plant cells lose water when they have been placed in a hypotonic solution, and therefore they become flaccid. So that process is plasmolysis. So the good students who did the work, I believe you have now understood it better. And to the other students who never did the work, like I said, we'll keep on praying for you. Great. So having seen this, now if you look at the diagrams here, this cell, the final cell that you can see here, is a bit smaller. It is relatively smaller as compared to these two other cells here. This one is very small. It is small because it has lost some of the water. And if you look at it, look at the shape, it has lost the shape. So a plasmolized cell does not, it loses the shape because it has lost the water, which made it turgid or gave it that shape that it had. And I think this, it's also explained when we were looking at this topic called transport in plants and animals, importance of water in the topic transport in plants and animals, transport in plants, sorry. We said that water helps in making the cells turgid, which brings about support in plants, okay? So, okay. So when we talk about plasmolysis is the process by which the plant cells will lose water when they are placed in a hypotonic solution, and then they become flaccid. Now, plasmolysis, the opposite of the word plasmolysis is deplasmolysis. Now, when a plasmolysed cell is placed in a hypotonic solution. Now, this cell, the cell that you're seeing here, when it is placed in this solution, which is hypotonic, it will gain water by osmosis, or it will draw water from the hypotonic solution by osmosis, and then it becomes turgid. So that the process of reversing plasmolysis is called deplasmolysis. So deplasmolysis is the opposite of plasmolysis. I'm explaining that because a student by the name of Carissa asked me to explain what deplasmolysis is, and that is also to the benefit of those people who did not know what deplasmolysis is. So up to that point, we proceed and look at these terminologies, which I said I will explain. That is turgor pressure and wall pressure. But before that, just to summarize what I've explained, we have said that when a plant cell is placed in a hypotonic solution or distilled water, it goes in water by osmosis and swells, but does not burst like in an animal cell. So when a plant cell is placed in a hypotonic solution or distilled water, it draws in water by osmosis and swells, but it does not burst. It does not burst. Now, this is because of the cell wall, which is rigid and prevents cells from bursting. This is because of the presence of the cell wall, which is rigid and prevents the plant cells from bursting. Now, I am trying to delay a bit because I know there are some students who are making some short notes. Okay. Now, turgor pressure. Turgor pressure is the pressure exerted by the cell contents against the cell wall when a plant cell is placed in distilled water or in a or in a hypotonic solution, I'll repeat that again. Turgid pressure is the pressure exerted by the cell contents against the cell wall. That pressure, that force that is developed by the cell contents as they push towards the cell wall is called the turgid pressure. And this is only seen when a plant cell is placed in distilled water or a hypotonic solution. And I said that as this happens, the cell will develop some force or pressure which acts in an opposite direction. So if the turgid pressure was pushing towards the right, the wall pressure will be pushing towards the left. And remember, the wall pressure is equal to the turgid pressure. So these forces that are acting towards the opposite direction, they make the cell to be very, very turgid, to be very strong, okay? To be very strong. So talking about wall pressure, this is the pressure that a plant cell exerts on the cell wall to counter or to balance or to resist the turgid pressure. It is the pressure that a plant cell exerts on the cell wall to counter or to balance or to resist turgid pressure. So when the wall pressure equals the turgid pressure, no more water can enter into the plant cell. When the wall pressure equals the turgid pressure, no more water can enter the plant cell. So the plant cell is said to be turgid. It is said to be turgid. Something that is turgid is very, very strong. So the plant cell is said to be turgid, or the cell becomes firm or rigid. The cell becomes firm or rigid. So this is what brings about support in plants. This is what brings about support in plants. This brings about support in plants. So more of this, like I said, was explained or was taught to you when we were looking at transport in plants and animals. And hopefully we'll be able to look at the topic again when we are done with nutrition in plants and animals, which is our next topic. Okay, so let us move and look at the next phenomenon that is related to osmosis, and this is wilting. Wilting. So wilting is a phenomenon that usually takes place in plants. Now, I believe before we proceed to wilting, there is no one with a question on water relations in plants, cells, and animal cells. If there is, please just let me know. If there is, just let me know. Meanwhile, let us look at wilting. Now, we know that wilting is a process or is a phenomenon that usually occurs in the plants. And let me just give you a highlight of what wilting is. We know that plants obtain water from the soil. Plants do obtain water from the soil. So once they obtain the water is used mainly for the process of photosynthesis, but it also serves other importances, like we've seen it provides support to the cells and making the plants to be very strong. But then this water, when it is in excess, it is usually lost through the process of transpiration. Okay? It is usually lost through the process of transpiration. Now, transpiration is a very active process. Transpiration is a very active process that takes place in the plants. And transpiration is a process by which plants lose water in form of water vapor. So, when plants lose water in form of water vapor, this water is usually replaced through absorption of water from the soil. So the plants will absorb water from the soil. They keep on absorbing water from the soil, but then this water is lost through transpiration. So you find that the rate of transpiration is usually very high during the day. The rate of transpiration is very high during the day, especially during the hot days, hot and dry days. When there is hot and dry conditions, the rate of transpiration is usually very, very high. So it gets to a point where the amount of water that is lost exceeds or becomes more than the amount of water that is absorbed by the plants. So because of that, the plants the plant cells become they become flaccid. And because of that, you'll be able to see that the plants now, they start drooping. They start drooping. Now the leaves and the branches will not be like very straight, but then they start behaving like they're about to fall. They start looking, they start facing downwards. Let me use facing downwards. I don't know the right term to use there, but I think that explains it better. They start looking downwards, they start facing downwards. So that brings about what we describe as permanent wilting. So the leaves after some time will start falling, the leaves will start falling, and then the plant will eventually remain with very few leaves, or in some cases, it will be having no leaves at all. And this is something that we've always seen during the dry seasons. So wilting occurs when the rate of water loss through transpiration, it occurs when the rate of water loss through transpiration in plants is more than the rate of water absorption from the soil. Now the cells, therefore, they develop reduced turgidity, and the plant droops. So we know that osmosis helps in the absorption of water from the soil. So when the plants absorb water, just like I said, this water is used for some important processes within the plants. It is used for some important processes within the plants, and for that matter, it is apart from the processes, the water that is in excess is usually lost through the process of transpiration. Now, we've said that when the process of transpiration is more than the rate of water absorption from the soil, that brings about wilting. So it leads to reduced turgidity, and then the plants will start drooping. Okay? Are we together up to that point? Great. So this phenomenon is called wilting, and it is observed in plants during hot and dry conditions. If the plant is well watered, the cells become turgid, and the turgidity of the cells is restored, and it grows upright again. So if you want to restore the lost turgidity of the cells, then just need to water the plants. That is why we always say that it is important for us to water, especially the young plants. When you grow young plants which do not have well-developed extensive roots that can be able to absorb water from different locations, then it is important to keep on watering them so that they absorb more water, even as active transpiration takes place within them. Okay? So we are saying that if the plant is well watered, cell turgidity is restored and it grows upright again. Okay. Now, but if water supply is inadequate, if there is inadequate water supply, the plant does not recover from wilting, and it is said to have undergone what we call permanent wilting. Permanent wilting. Remember, transpiration will always take place. It never stops. It will only stop when the plant has completely lost all its water, and there are no more leaves to take place to help the process of transpiration. So if there is inadequate water supply, the plant cells do not recover from wilting, and then these plant cells this leads to permanent wilting in plants. Now, wilting has an advantage to plants. It is not just a disadvantage, but in some cases it has an advantage to plants because it helps them to conserve water by reducing transpiration, because the drooping leaves expose less surface to the sun and wind, which are very strong agents of transpiration. So, I mean, strong factors that increases the rate of transpiration, sorry. So wilting helps the plants. It helps the plants to reduce transpiration, thus conserving the amount of water that is present for other important processes within the plant. So that is why I'm saying here that wilting has an advantage to plants because it helps them to conserve water by reducing transpiration. Because the drooping leaves, they face downwards, remember? They do not expose a large surface area, but they expose just a less surface area to the sun and wind, which usually increases the rate of transpiration. So when we have drooping leaves, just a small surface area is exposed to the sun and thus that reduces the rate of transpiration. Great. So let us very first look at the roles of osmosis and the factors affecting the rate of osmosis before we look at the last process, which is active transport. So I will very first look at the roles of osmosis and factors affecting the rate of osmosis in organisms. So let us first start by looking at the roles of osmosis in organisms. The roles of osmosis in organisms. Number one, osmosis plays a very important role in support in herbaceous plants. It plays a very important role in support in herbaceous plants. And this is explained that now when plant cells take in water by osmosis, when the plant cells take in water by osmosis, like we have seen, the cells become turgid. The cells become turgid. And know that the whole structure of a plant is made up of the cells. We have got plant cells in almost all parts of a plant. So when all the cells within a plant become turgid, they bring about support. They bring about support. So this turgidity gives support to seedlings, the young plants, to the leaves, and to the less woody plants like the herbaceous plants. Now, herbaceous plants, you know, they are less woody. They are less woody. So the turgidity of the cells brings about support to the seedlings, to the leaves, and to the less woody plants like the herbaceous plants. Now, support is just brought about by the turgidity of the cells. When the cells become turgid, they make the plant to be very strong. Okay, so that turgidity is what brings about support. So support in herbaceous plants. Number two, water absorption from the soil by the plant roots. Water absorption from the soil by the plant roots. Now, we know that if you look at the structure of the roots, we have got those extensions, which you call the root hairs. The root hairs are the ones which are usually useful when it comes to absorption of water from the soil with the process of, with the help of osmosis. So we think that plants absorb water from the soil by osmosis due to the high solid concentrations in the cell sap of the root hairs, the root hair cells. So the cell sap of the root hair cells have got a high solute concentration than that in the soil. So because of that concentration gradient, there'll be that net movement of water molecules from the soil through the cell membranes of the root hair cells into the cell saps of these root hair cells. So that brings about, you can now see that is what contributes to absorption. As you can see, water molecules moving from the soil where they're highly concentrated into the cell sap of the root hair cells where there is low concentration of water. Okay. The next role of osmosis that I want us to look at is opening and closing of the stomata. Opening and closing of the stomata. Now, this is better explained in the topic gaseous exchange, which I think some of the teachers introduced into their classes. But I just want to give a highlight. Opening and closing of the stomata. Now, stomata, as we all know, stomata are those external openings that are found on the surface of the leaves, usually in the epidermis, in the epidermal cells, either on the upper epidermal cells or on the lower epidermal cells. So we have got the stomata there. The stomata are usually surrounded by the guard cells. They're usually surrounded by the guard cells, and the guard cells contain chloroplasts. The guard cells contain chloroplasts, which actively take, the chloroplasts which we know take part in photosynthesis. They are the ones that that performs the function of photosynthesis in plants. So because photosynthesis, as we know, is the process that leads to the formation of simple sugars, and simple sugars such as glucose, which are osmotically active. In other words, when we have got glucose within the cells, and not just in the cell, but within the guard cells. And I want you to get that point. I started by saying that, and I know I'm a bit fast. I will try and explain this. Stomata, we are looking at the function or the role of osmosis in opening and closing of the stomata. Okay? Stomata are found on the epidermal cells. And stomata are always surrounded by cells called the guard cells. The functions of the guard cells is to control the opening and closing of the stomata. In other words, the guard cells can either allow the stomata to open or to close. Okay? The guard cells contain organelles called the chloroplasts. Chloroplasts help in the process of photosynthesis. When photosynthesis takes place, or when the cells, when the plants photosynthesize, they produce foods in form of simple sugars, and these simple sugars we call them glucose. Now, glucose is osmotically active. In other words, glucose has the ability to draw water from the adjacent or neighboring cells. So remember, glucose is now formed inside the guard cells because that is where we have the chloroplasts. This will therefore make the guard cells to draw water from the surrounding cells, which is the epidermal cells here. From the surrounding cells, which is the epidermal cells. Okay? So when water is drawn into the guard cells become turgid. This makes the thick inner walls of the guard cells to distend or to move away from one another, while the thin outer walls of the guard cells will bend, or they will also enlarge. They will swell, and that leads to the opening of the stomata. Okay, and usually this happens during the day when the process of photosynthesis is actively taking place because of the presence of the light from the sun. We know that at night photosynthesis does not take place, but instead an active process of respiration, which we also know takes place during the day and at night. But at night, usually the rate of respiration is high. So plants, the plants are usually respiring more at night. So at night, when there is no photosynthesis, we have got the glucose that are usually formed. They are converted to starch. Okay, they are converted to starch, and starch is osmotically inactive. So when we have starch inside the guard cells, starch, like I said, starch is osmotically inactive. In other words, it does not draw water. It does not draw water from the adjacent cells, but instead it makes the cells, the guard cells, to lose water to the surrounding epidermal cells. So water is now lost from the guard cells back to the surrounding epidermal cells. And so when the plants lose the water, when the guard cells lose the water back to the epidermal cells, they become less turgid. They become less turgid, or they become flaccid. When they become flaccid. So when they become flaccid, the stomata closes. So the guard cells, they pull towards each other, and then the stomata closes. So I know some of you may not have understood clearly how osmosis leads to the opening and closing of the stomata, but I know it is more of this is covered in the topic gaseous exchange, which is in Form 2. So there is a whole theory which is called the photosynthetic theory, which explains the opening and closing of stomata and how osmosis leads to that. So do not worry, because more of this is explained there. But again, if you are a good student and you don't understand this, at your own free time, you can look at it, or you can ask your teachers, or you can ask me. I'll be able to respond to that, okay? So we are saying that, let me just a summary of what I've said. We are saying that when guard cells gain water by osmosis from adjacent epidermal cells, they become turgid. They bulge outwards, and the stomata opens. When the guard cells lose water to the surrounding epidermal cells, they become flaccid, pulls towards each other, and the stomata close. Okay, and the stomata close. Great. So that is how osmosis leads to the opening and closing of the stomata. The fourth role that is played by osmosis is feeding in insectivorous plants, feeding in insectivorous plants. Sorry for that. Feeding in insectivorous plants. Now we have got these plants which are called insectivorous plants, and I want you to find out some of the examples. Find out some of the examples of insectivorous plants. At your own free time, please find out some of the examples of insectivorous plants, and you can let us know in our next class. So why do we call them insectivorous plants? Now, these insectivorous plants, they are plants that have got the ability. They have the ability to trap and feed on insects, certain insects, where they are able to obtain nitrogen, which they use to manufacture proteins in them. So the reason why they trap and feed on these insects is so that they can be able to acquire nitrogen, which is useful in the manufacture of proteins. Plants really need the proteins. So this group of plants, which we call the insectivorous plants, they obtain nitrogen by trapping and feeding on these insects. Okay? So how is osmosis as a factor now used here? We are going to see that. We are saying that these are plants that trap and feed on insects to obtain their supply of nitrogen, which is lacking in the soils in which they grow. So you can see that these insectivorous plants, the reason why they're doing this is because they do not obtain nitrogen from the soil that they grow. And therefore, the only way they can obtain the nitrogen is by trapping and feeding on these insects, okay? So change in turgid pressure in special structures. Now these plants, they have got special structures in which can detect change in turgid pressure. So we are saying that change in turgid pressure in special structures enables them to close, trapping the insects, which are then digested. So there is usually a change in turgid pressure, which is detected especially when insects land on those structures. So when the insects land on those structures, there is usually a change in turgid pressure. So this change in turgid pressure is usually detected, and then the plants respond by trapping, or they close the structures. And when they close the structures, they trap the insects, and these insects are finally digested, and they produce nitrogen, which are used by the plants. If you haven't understood the point about feeding insectivorous plants, I'd advise that you read more about it. There's a lot of documentaries, there are a lot of videos online which can help you understand this. Okay. The fifth other importance of osmosis is water reabsorption in kidney tubules. This occurs by osmosis during the process of osmoregulation, and more of osmoregulation we'll look at when we'll be looking at the fourth topic. The fourth topic, which is excretion and homeostasis, which is in Form 2. We'll get to know how this happens. But just to mention, we know that osmosis helps or leads to osmoregulation, and this leads to water reabsorption in the kidney tubules. We also have water absorption from the intestines, and this is usually in the large intestines where there is usually absorption of water. So this occurs by osmosis, usually after digestion. So digestion we know digestion does not take place in the large intestines, but some foods are usually digested in the stomach, and then they're absorbed in the large intestine. So because there's usually that need for water to help in the transport of certain nutrients, then there is that absorption of water from the large intestines, and this is usually aided by the process of osmosis. And then we also have water movement from cell to cell, both in the plant and animal cell. Now, once absorption has taken place by the roots, let me explain this. Once absorption has taken place by the roots of plants, there is that movement of water from cell to cell until where it gets into the vascular bundles before it is finally transported into the leaves through the stems of plants. So that movement of water from one cell to the other is aided or is brought about by osmosis. When one cell is highly concentrated with water than the other cell, then there'll be movement of water from that cell to the other where there is low concentration. So water movement from cell to cell by osmosis. Okay. So there's just some important point to note here. We're saying that the osmotic effect is applied in food preservation, whereby bacteria are killed by surrounding them with highly concentrated sugar solutions, and that is always common in honey preservatives or highly concentrated salt solutions. Now, this point, I know you may not understand it, but just to get it from my point here, the osmotic effect is applied in food preservations, whereby bacteria are killed by surrounding them with highly concentrated sugar solutions or highly concentrated salt solutions. That leads to the killing of bacteria. And this technology is used in food preservation so that you keep the food free of bacteria. So the food are usually kept or surrounded by high concentrated solutions of sugar and salts. So these ones, why do we say this? When we have got when a bacteria is surrounded by a high concentration of either sugar or salt, this makes the bacteria to lose water by osmosis. They become dehydrated and then they die. Okay. So the next thing that I want us to look at very fast are the factors which affect the rate of osmosis. And I'll just mention this because I know they are very, we had looked at some of them when we were looking at we had looked at most of them when we were looking at the factors affecting the rate of diffusion. So I will just mention them because, as you can see, I've indicated here that factors that affect the rate of diffusion also affect the rate of osmosis, okay? Some of the factors that affect the rate of diffusion, such as concentration gradient, they also affect the rate of osmosis, okay? And these factors include, number one, we have concentration gradient. Concentration gradient, you can write them down because they are not on they're not part of my slides here. We have concentration gradient and concentration of solutions. Concentration gradients and concentration of solutions. And we also have another factor which is important here is temperature. Temperature. There is a hand that has been raised here by Ernest Mumu. Ernest Mumu, could you please ask the question?
Now, where I have said that the osmotic effect is applied in food preservation, whereby bacteria are killed by surrounding them with highly concentrated sugar solutions. Does it mean that by surrounding the food with the highly concentrated solutions, whether salty or sugary, does it also dry the food? Yes.
Okay. Thank you. I've heard your question. So this technology is okay. When you talk about the osmotic effect being used as a food preservative, it is just meant to keep the bacteria away from the food. So with the bacteria, when they get in concentrations of sugar. Okay, could you please mute yourself? Let me just mute you kindly. Okay, so when the bacteria gets into this food, which have been preserved using the high concentrated solutions of sugar or salt, the bacteria will lose the water which is found in their bodies because of the water will be drawn into the high concentrations of sugar and salts. And I believe you understand that. Do you understand that? Great. So if this happens, the
Yes, Ernest Momo. Momo, what's your problem? The question?
The teacher had not yet finished explaining it.
Oh, yes, just they had some network problem. Is coming back. What was the question?
I was asking
Mm.
if we use like highly concentrated salt or sugar solutions to preserve food, that is by drawing water from the bacteria by osmosis, does it mean that it will also draw water from the food? Let's say, for example, meat. Yes.
Mm. You see, when you add salt to meat, okay, are you getting me?
Yes.
Salt is an hygroscopic substance. That means it absorbs the water from those wet materials. So if meat is wet, and when you pour some salt on meat, you'll find that on the surface there will be some wetness, and therefore that salt will absorb the moisture that is present in that meat to make it to be dry. So when it is dry, the bacteria cannot survive in a dry environment. So it is not necessarily the bacteria that are being removed, the water is being extracted from them, but the food substrate. You know, the substrate is the meat here. Okay, so if the
Yes.
substrate is dry, the bacteria may not survive in such an environment. That is why if you apply salt on that, the salt will dehydrate the particular meat, and therefore it will become dry, and the bacteria may not survive. Also, when you have a wound, you're always advised to apply some salt. So application of some salt there make that place to be, so that the water can be withdrawn. It is dehydrated, and again it makes it become somehow alkaline in a way. You understand?
Yes.
Hello, Desmond.
Yes, Marlin.
Please continue. Oh.
Okay, thank you. Sorry, I lost my connection for a while, so I disappeared, but I'm now back, and let us continue. So, Ernest, I think Mwalimu has tried and answered the question. I just wanted to try and make you understand that.
sorry, Desmond. Just elaborate to him. I didn't know you were in.
Okay. Thank you, Mwalimu. I think you've said everything, plus even the chemist in it. So, okay, Mwalimu, we need to mute now.
Okay.
So, when Ernest, I think your question has been well explained by Mr. Onyango, and there'd be no need for us to continue talking about that. The most important concept, once you understand, now the foods that are usually preserved by this kind of technology, like I said, they in most cases dry foods, okay, dry foods, and these highly concentrated solutions of salt and sugars, they have got the ability to draw water from the bacteria. So of course, when bacteria comes into contact with such kind of foods, they'll not survive because they'll be dehydrated and finally die, and the food will be kept free from bacteria. Okay. So as we continue, I think I've given the explanations of some of the factors that affect the rate of osmosis, and I've said that we have got number one factor is temperature. Number two, we have got what we call concentration gradients, or the concentration of solutions. You notice because of the level of concentrated of solutions that we have either hypertonic or hypotonic solutions. Okay. So when we talk about temperature as a factor that affects the rate of osmosis, remember, osmosis takes place or usually happens across the cell membranes, okay? And cell membranes are made up of proteins. So high temperatures will denature or will interfere the structures of the cell membranes, and therefore will affect the movement of water molecules across the cell membrane, okay? While I think there's an urgent question here. Oh, okay. So someone is asking, does pH affect osmosis? I don't. pH does not affect osmosis. Yeah, it doesn't really affect osmosis. It is just temperature which affects the structure of the cell membrane, and you know that osmosis occurs across the cell membrane. When we talk about the concentration solutions or concentration gradient, the different when we have got a steep or a high difference, a high concentration gradient, then the rate of osmosis will be very fast as compared to when you have a small or a low concentration gradient. The same happens when talking about concentration of solutions. When we have a solution that is highly concentrated and one that is lowly concentrated, then there'll be movement of water molecules from the low concentration. There'll be a high movement or a high rate of movement of water molecules from the lowly concentrated solution to the highly concentrated solution. Okay? Yeah, so those two factors are the ones that affect the rate of osmosis. Okay. Okay, someone is just is reminding me that pH affects the permeability of the cell membrane. I want to say that the pH does not affect the permeability of the membrane. What the pH does, we know that the cell membrane, it has got both the positive and the negative charges. Okay, this affects the movement of substances. It affects the movement of substances. Now, the one that affects the permeability here is temperature. Temperature affects the permeability because you know the membrane is made up of proteins. Okay, and these proteins can be affected in extreme temperatures. I remember I gave an example and I mentioned enzymes, which are protein in nature. And you know that these enzymes, they can be denatured or be made inactive in certain ranges of temperatures. So when the temperatures are high, the enzymes are denatured. When the temperatures are low, the enzymes become inactive. Okay? Okay. So someone by the name David Gaichema, I just want to make this point clear, says that, teacher, we were taught that since membrane is proteinous in nature, pH affects movement of substances, which is by osmosis. pH affects movement of substances, which is by osmosis. Now, movement of substances. Now, you know that it is all about the polarization of these substances. You see, movement is totally different from the structure. Now, those who can't hear me, I think it's a problem with your connections, not mine. I'm a bit clear to a good number of students. So Victor and Jega, you need to work on your connection. So movement is movement. Leave alone the structure. So when the structure is interfered with, then it means there'll be no movement. Okay? So polarization is brought about by the ability of the membrane to detect changes in pH. So I think I made this point clearly. Do not confuse the structure and what brings about movement. So movement is that act of passing when the substances are passing across the membrane, and this can be as a result of the charges, because, and I think we all know that there is this principle that is always, it's common, I think, in physics, whereby you talk about the like charges attract and like charges repel. So we didn't say that the membrane, okay, we said the membranes are protein in nature, right? They are made up of proteins. But then we said the point about pH was the membranes, they have got the ability to detect changes in temperature and pH. They can detect changes in temperature and pH. And pH here, the fact the pH as a factor here affects the movement of substances. Temperature, when we have got high temperatures will structure of the membrane because of the proteins which are part of the membrane. I don't know that I'm making myself clear, students. Now, I want to challenge those students who are not understanding the points about pH and temperature as a factor, which are properties of the cell membrane, to go and read more about them, because it's like you are taking as bad, which is not bad. But could you please just take your time and read about the properties of the cell membrane, students? So. Then before I continue, there's a student who is saying, okay, I said, Victor, if you couldn't really hear me, then work on your connection. It is not mine. Mine is better. Then someone is asking my number. I will give my number at the end of the class. Then diffusion and active transport. Diffusion and active transport. I don't know what the question is, Lewis, but we are just about to look at active transport, okay? And that is the next thing that I'm looking at here. So if you still find the problem with understanding how pH and temperature affects the membrane, then please look at them. Read them. It is not just a matter of looking at them, but try and understand. So. Yeah. Active transport is our next and our final, is our last physiological process. And once we're done with active transport, we'll be able to finish the topic. So we've been talking about active diffusion and osmosis. And we said that these are processes that help in the movement of substances from one region to the other. Now, we know that there are certain substances which are also important to the cells because they are required for some processes to take place. But because of their sizes, they are unable to move freely. Now, I want to give a good example of the mineral ions which are usually present in the soil and they are required by the plants. They are present in the soil, but they're required by the plants. Okay? So when these mineral ions, because of their sizes, one, they may not be easily allowed to pass through the membrane. They're different from gases and liquids which can easily pass across the membrane. So for the mineral ions to leave the soil and finally get into the plants, they'll still have to pass by the membrane. And so the membranes, that transport, movement of substance across the cell membrane can sometimes be aided by substances that we call carriers, okay? They can sometimes be aided by some substances that we call the carriers. Now, the carriers will combine with these ions, which are required to pass across the cell membrane so that they are finally carried or transported across the membrane into the cell. So carriers are usually found on the cell membrane. And these carriers, for them to transport these substances, they require some energy. Okay? They require some energy so that they can help in moving these substances across the membrane. Okay? Seems like there are a lot of questions. Okay, are you able to hear me? Because there's one student who is saying that he can't hear me. Can everyone hear me? Just let me know. Newton Buru, can you hear me? Yeah, I can see him nodding. He said that means he can hear me. Let me see if another student here can hear me, because I'm worried Newton Victor Njera can't hear me. Moses Carissa, can you hear me? Gerald Muoki, can you hear me? Good. So kindly, Victor Njega, you need to work on your connection, because it's like you are the one who has the problem. Just work on it kindly. Okay. A question from Jack Mutuma. Oh, Jack Mutuma, you lowered your hand, okay? Okay, so we can continue. Now we're looking at active transport. Now, active transport can be defined as the process. Active transport is the process by which molecules and ions move from an area of low concentration or a region of low concentration to an area or region of high concentration across a semi-permeable, across a cell membrane. And you can see now this movement that takes place against a concentration gradient. Now, against a concentration gradient means it is coming from a low concentration to a high concentration. These are different type of, it's a unique physiological process, because if you compare to the others, you saw movement of substances from a region of high concentration to a region of low concentration. But here, we're talking about movement of substances from a region of low concentration to a region of high concentration. So this is against a concentration gradient, okay? So because there is that movement against a concentration gradient, it is literally difficult for molecules and ions to move from a region of low concentration to a region of high concentration. And so this movement is aided by what we call carriers, okay? Carriers. Now, carriers are substances that combine with the molecules and ions that are being transported, and then they help them to move across the membrane against the concentration gradient. And because of the carriers here, this process requires the use of energy. This process requires the use of energy. Now, you can be asked to define the meaning of the word active transport. The meaning of the word active transport. The most important thing that you should mention here is the movement of molecules and ions from an area of low concentration to an area of high concentration across a cell membrane. Now, some people would add the word use of energy in the definition. It is not wrong, it is right. Of energy, it's right. You can add that, or you cannot add. You won't be penalized for not using the word energy there. Okay, so it is an active, from the definition, it is an active process. It is an active process. That is why we call it active transport. It requires the use of those carriers which combine with the ions and molecules before they are transported. Okay. Yeah. So in active transport, substances move against a concentration gradient. And this process requires the use of energy, okay? So we're saying that it involves protein carrier molecules that combine with the substance being transported and then move them from one side of the membrane to the other. So you understand how these protein carriers work. They combine, the carriers are protein in nature. They combine with the substances which are to be transported. Then they move them across the membrane from one side to the other. Are we together? So you can see this. You can see these are diagrammatic presentation of what we're talking about. We have got the membrane. This space here is the membrane. Then we have got one side of the membrane and the other side of the membrane. One side of the membrane has got a low salt concentration, and the other side of the membrane has got a high salt concentration. Okay, salts are made of ions. So as you can see, salt is moving. There is that movement of salt from the region where there is low concentration to the region where there is high concentration. So on the surface of the membrane here, we have what is called protein carrier molecule. And the protein carrier molecule, as you can see, it combines with the ions. It combines with the salt ions, and then it transports them across the membrane to the region where there is high concentration. It transports them to the region where there is a high concentration, okay? I think this diagram here has helped us to understand what we mean when talking about the protein carriers, okay? So what are some of the roles of active transport in organisms? Roles of active transport in organisms. There are several roles of active transport in organisms, and they include: number one, Absorption of mineral salts or ions from the soil by root hair cells in plants. Remember, mineral ions are lowly concentrated in the soil as compared to the cell subs of the plant cells. So there is that movement of the ions from the soil where they're lowly concentrated to the cell subs where they're highly concentrated. So we're talking about absorption of mineral ions from the soil by hair cells in the plants. Number two, and I believe you are writing these short points for your own revision, absorption of digested food from the alimentary canal into the bloodstream in animals, absorption of digested foods from the alimentary canal in the bloodstream, into the bloodstream in animals. Then we have reabsorption of sugars and some salts from the kidney tubules back into the blood. This will be able to look at it in the topic excretion and excretion and homeostasis, a very, very interesting topic in Form 2. Just note it is used, active transport is used in the reabsorption of sugars and some salts from the kidney tubules back into the blood, okay? Number 4, removal or excretion of metabolic waste products from body cells. Removal or excretion of metabolic waste products from body cells. Number 5, accumulation of salts or solids in the body cells to offset osmotic imbalances in organisms that live in arid and saline environments. You could just write the points. If you are cut short of time, I can be able to explain them in our next class because I only have 4 minutes left. I can be able to explain this in our next class. So just make sure you have the points. Do not leave any behind. So we're talking about accumulation of salts or solutes in the body cells to offset osmotic imbalances in organisms that live in arid and saline environments. Saline environments are very salty environments. And once again, this is also another thing that you'll be able to understand when we'll be looking at the topic excretion and homeostasis that is in Form 2, okay? And then number 6, we have got what we call transmission of nerve impulses in the nerve cells. Transmission of nerve impulses in the nerve cells. That is also aided by active transport. So there is active transport where there is that transmission of nerve impulses in the nerve cells. This is a topic in Form 4. Yeah, a topic in Form 4, but just mention the points. So, yeah, and then finally we look at the factors affecting the rate of active transport. Factors affecting the rate of active transport. Number 1, we said that because active transport is a process, is an active process, and it requires the use of energy. Active transport is an active process that occurs in the cells, and it requires the use of energy. So it means that the process of respiration is very key here. The process of respiration is very key because it is respiration that leads to the formation or to the production of energy. And so those factors that affect energy production in the cell through respiration, they also affect the rate of active transport. And they include, number 1, oxygen concentration. Oxygen concentration. Oxygen is very, very important in the process of respiration because the process of respiration, we know that it can take place both in the presence or in the absence of oxygen. But usually, when it happens in the presence of oxygen, there is a lot of energy that is produced. So when the concentration of oxygen is high, when the oxygen concentration is high, the cell produces more energy, which allows for the rapid active transport by the protein carrier molecules. Those carrier molecules require energy. The protein carrier molecules require energy. And when there is high oxygen concentration, it means a lot of energy will be produced, which will allow for rapid active transport by the protein carrier molecules. Okay? The next factor that affects the rate of active transport is glucose concentration. Glucose concentration. Now, glucose here is a substrate that is used to produce energy. Glucose is a substrate that is used to produce energy. And so, when we have got a high glucose concentration, when we have a high glucose concentration, when the amount of glucose is available is high, then it means that the amount of energy that produced will also be high. And that is why I've said on my there that the amount of energy produced by a cell depends on the amount of glucose available. Okay? The amount of energy produced by a cell depends on the amount of glucose available. All right? Then the amount of glucose available, the more the glucose available, the more energy produced, hence the faster the rate of active transport, and vice versa. The more the glucose available, the more the energy produced, hence the faster the rate of active transport, and vice versa. Okay? We also have temperature, and you say that high temperatures, or usually temperatures which are above 40 degrees, will denature or will destroy the protein carrier molecules. The carrier molecules that, like I said, are protein in nature, and they can be denatured or destroyed by high temperatures. Okay? So high temperatures, which are usually above 40 degrees, will denature or destroy the protein carrier molecules in the cell membrane. And this therefore will stop active transport from taking place. So when these protein carrier molecules are destroyed, active transport will be stopped. Okay? Another factor, my time is already up. Another factor that affects the rate of active transport is pH. pH. So pH, extreme change in the pH of the surroundings of a cell, they alters the structure of the protein carrier molecules. Extreme changes in pH in the surrounding of a cell alters the structure of the protein carrier molecule. And this therefore reduces the ability of these protein molecules. This reduces the ability of the protein carrier molecules to actively transport molecules across the cell membrane. To actively transport molecules across the cell membrane. Okay? And then we have another factor called the enzyme inhibitors. Enzyme inhibitors. Now, an enzyme, as I said last time, is a substance that can either speed up the rate of a reaction or it can lower the rate of a reaction. So enzymes, enzyme inhibitors, they make the enzymes less inactive or they make them inactive. They can make them less effective or they can make them inactive. Now, enzymes. These protein carrier molecules, it's like they act almost like enzymes here because they're the ones that facilitate the process of active transport. They like carry molecules from one place to the other. So, in the presence of these enzyme inhibitors, respiratory enzymes, which are useful, which increases the rate of respiration. Allow me to repeat that point. In the presence of inhibitors, respiratory enzymes. Now, the process of respiration is facilitated by what we call the respiratory enzymes, okay? So these respiratory enzymes, which are involved in the release of energy, they become less effective or inactive. And less or no energy can therefore be produced. So when there is less or no energy produced, then there'll be no active transport taking place. So when in the presence of enzyme inhibitors, the respiratory enzymes will not, my screen has frozen. Okay. Mr. Onyango, can you hear me?
Yes, we can still see the screen.
Oh, okay, okay. Thank you, Malim. Now, in the presence of the enzyme inhibitors, respiratory enzymes, now those enzymes which help in the process of respiration, they may become less effective or inactive. And when they're inactive or less effective, it means that either less energy will be produced or no energy or no energy at all. So this prevents active transport from taking place. Okay. Let's proceed and try and beat time. Okay, that marks the end of my slides and marks the end of the topic. We are stopping at that point, having looked at the active transport, roles of active transport, and the factors affecting the rate of active transport. Now, for those who haven't understood, because I know I've looked at active transport very fast, could you please channel your questions or concerns? You can channel them. Like I said, I will give my contact to those who wanted them. My number is 0751 0751 91 0004 0751 91 0004. Okay, so if there are questions, you can ask them through that number. I'm available on WhatsApp, or we can ask them in our next class, that is on Monday next week, and you will be able to get all the responses that are needed that you want. Okay.
Hello? Hello, Desmond.
Yes.
You said the number is 07?
0751
0751
91
91
0004.
0751. Oh, we the student now. Ni sawa, I think wamepata. Now,
Okay.
to the students, Mr. Desmond is really doing some good job, and always feel free to inbox him. You can have some personal classes with him. I'm wondering, some of you are just coming in, they leave before the lesson ends. When you have such committed teachers, it's very a rare opportunity. Some schools are really longing to have such. But if you can't utilize whatever you're given there, I'm wondering which type of form tools we are. Some of you are around 190 now. I'm only seeing 113 a lesson Aisha. Don't come to class because your parents said you come. I'm very grateful for those of us who have stayed up to now. Let us always, if you want those presentations, those PowerPoint notes, always lies Mr. Desmond. He will always help you. You can even have some private classes with him. Just organize with him. You can. He'll be a great deal for you. Be a very great deal for you. He's a good man. He has done well. I enjoyed the presentation. The class was so nice. I've learned a lot. Repeat the number. 07 Desmond 0750.
0751
0751
91
91
0004.
4. I think I've sent it in the inbox. Everybody can see it there. Please, if you want to contact Mr. Desmond, do that. You can have some private lesson with him. He's very flexible. I think he will help you a great deal. Ama mwalimu si ni kweli?
Yeah, I'm always available, Mwalimu. Thank you.
So you guys have a good day. Prepare for the next class and always be cooperative. Advise those who are not coming to come in at all times. Have a good day.
Thank you and have a good day, Mwalimu.